righty. Good morning. Good morning. Almost everybody's here. Wonderful on our last day before Thanksgiving break. Yay. All right. Everybody, while we're waiting for everyone, a couple of more people coming in, look at your Canvas page for, let me get there first. Technology, DNA technology. We're going to do an activity. I want you to read over that page first. Okay, so if everybody would go to Canvas, DNA technology lesson. Take it over that page for a few minutes while we're waiting for people to come in, and then I will run through the activity with you. All right, for those that just came in, good morning. We're in Canvas. We're reading over the DNA technology lesson. All right. All right, so you read over the parts that need reading, and then I will watch the two videos together, and then I will share with you what we're going to do with it. I just wanted to make sure the sound part was on, which it is. Okay. Mm, okay, we had somebody in the wrong class. All right, good morning. We're reading the Canvas page on technology. Quickly do the attendance and then I'll share back. All right. If you haven't looked at that box, we'll come back to that because I want to highlight that as part of the activity that you're going to do. So I'm going to go ahead and play these two videos for you, and then we'll come back and talk about those.
Using genetic engineering, scientists for example, circular pieces of DNA called plasmids inside bacterial cells to make such proteins as human insulin by inserting the gene that codes for that protein. Special enzymes called restriction enzymes, which recognize, bind to, and cut specific DNA sequences, are used to open up a plasmid in preparation for the insertion of a gene. Some restriction enzymes leave the DNA with uneven ends, also known as sticky ends, shown here. If the gene we wish to insert and the plasmid are both cut with the same enzyme, these sticky ends will be complementary, meaning they will readily bond with one another. In a follow-up reaction, gene fragments, cut plasmids, and an enzyme that connects DNA fragments are mixed together. This so-called ligase enzyme attaches the sticky ends in the solution to produce circular plasmids that now carry the gene that codes for the desired protein. Note that the efficiency of recombinant techniques is not 100% and may lead to plasmids with no inserts or multiple inserts.
Okay. So just take note that that one was called recombinant techniques. All right, let's look at another one.
The ability to create a technique to clone human beings has been ongoing. Based on all the discussion, it's an easy process. What is actually involved by viewing our short animation. What are clones? Simply put, clones are organisms that have identical genetic material. In other words, the sequence of bases in their DNA is exactly the same.
Excuse me, you're not sharing your screen.
Oh, what happened to it? Okay. Thank you. Sorry. It was sharing and then it stopped. All right. Okay, wait a minute. Let me stop this. All right. Did you see the first one? And thank you for coming off mute because I was so busy watching the screen that I didn't see you. So you didn't see the first one either. Okay, so let me go back and do that one again. So let's look at this one. Sorry about that. I had it up and then it dropped down and I think I moved the chat box. Thank you, Isabella. It's Friday. Oh, okay. There we go. All right. Before, because I was taking notes on it. Um, before I let this one play, you also jot down some notes about the video because the project that you're going to do is want you to select a type of technology. So we're going to be looking at this technology and so the cloning and all of these things that they're doing with DNA these days. And you're going to pick one. So just be listening to all the different things that are available.
Using genetic engineering, scientists can create cells that produce proteins they wouldn't ordinarily produce. For example, circular pieces of DNA, called plasmids, found inside bacterial cells, can be engineered to make such proteins as human insulin by inserting the gene that codes for that protein. Special enzymes called restriction enzymes, which recognize, bind to, and cut specific DNA sequences, are used to open up a plasmid in preparation for the insertion of a gene. Some restriction enzymes leave the DNA with uneven ends, also known as sticky ends, shown here. If the gene we wish to insert and the plasmid are both cut with the same enzyme, these sticky ends will be complementary, meaning they will readily bond with one another. In a follow-up reaction, gene fragments, cut plasmids, and an enzyme that connects DNA fragments are mixed together. This so-called ligase enzyme attaches the sticky ends in the solution to produce circular plasmids that now carry the gene that codes for the desired protein. Note that the efficiency of recombinant techniques is not 100% and may lead to plasmids with no inserts or multiple inserts.
Okay, so that technology is recombinant DNA technology. So just take a note of that one. And now we're going to go back and look at cloning. So let me get this one to start back from the beginning.
Cloning 101. The ability to create a clone used to be science fiction. Dr. Ian Wilmot's group changed that in 1997 with the creation of Dolly the sheep. Since then, the debate on applying the technique to clone human beings has been ongoing. Based on all the discussion, it may sound like cloning, human or otherwise, is an easy process. Find out what is actually involved in the cloning process by viewing our short animation. What are clones? Simply put, clones are organisms that have identical genetic material. In other words, the sequence of bases in their DNA is exactly the same. Long before the birth of Dolly the sheep, clones had been observed in both nature and in the laboratory. When a couple has identical twins, or triplets for that matter, the children are clones of one another. A plant cutting can also be used to generate a clone. Prior to 1996, it was thought that cloning an entire animal could only be done with embryonic cells, cells present in the early stages of an organism's development. In the 1950s, scientists generated entire frogs from embryonic frog cells. After a small number of cell divisions, embryonic cells start to change into different types of cells that an organism needs, including cells that form muscle, blood, liver, etc. This process is called differentiation. Although each of these cells has the same genetic material, each cell can only access the genes needed for its particular function. Before the experiment at the Roslin Institute, it was thought that once cells differentiated, they could not be used to generate an entire organism. For instance, in a sheep, udder cells could generate other udder cells, but not an entire sheep. The scientists at the Roslin Institute solved this problem by growing sheep udder cells under starvation conditions. This put the cells in a state similar to embryonic cells. This is called the G0 state. An egg cell was taken from another sheep. The nucleus, which contains the genetic material, was removed from the egg cell using a very fine needle. They then used electric shock to fuse one starved udder cell with one nucleus-free egg cell. They made 277 of these fused cells. Although the egg cell came from a black-faced sheep, notice that the nucleus with the genetic material came from the white-faced sheep. These fused egg cells were then inserted into several different sheep. These surrogate mothers were also black-faced. Of the 277 fused cells, only one progressed to form a developed lamb. Dolly was born on July 5, 1996. Scientists found that Dolly had the same DNA as the udder cells she came from. She is a clone of these udder cells. Dolly has given birth to a lamb named Bonnie, produced the natural way. Other lambs have been born at the Roslin Institute through their cloning process. Some carry genes that will produce usable human drugs. A laboratory in Hawaii, run by Dr. Ryuzo Yanagamachi, was the second group to successfully clone an animal from an adult cell. They cloned mice using cumulus cells, a cell type found in the ovaries. The cloning method used by the lab in Hawaii was different in two ways from the method used to clone Dolly. First, the cells used to clone the mice were not grown in culture, but instead were used immediately. Second, the nucleus was removed from the cumulus cell and then directly injected into the egg cell. This egg cell's nucleus had already been removed. The Yanagamachi lab used coat color to track genetic heritage. The cumulus cell comes from an agouti, brown, mouse, and the egg cell comes from a black mouse. The egg cell now had the same genetic information as the nucleus donor mouse. The egg cell was then activated and implanted into a white host mother. On October 3, 1997, the host mouse gave birth to Cumulina, named after the cumulus cells she was cloned from. Cumulina is the same color as the mouse that donated the nucleus. DNA fingerprinting confirmed that Cumulina had the same DNA as the nucleus donor. The scientists have taken cells from Cumulina to make more clones. They have successfully made several generations of clones, and all mice seem normal. Dolly the sheep died at the age of 6. Since the world said hello to Dolly, several other animals have also been cloned. Both Dolly and Cumulina were cloned from cells in the female reproductive system. Cows have also been cloned using ovary and cumulus cells, with the same method that was used to clone Dolly. Pigs have been added to the cloned animal menagerie. Scientists hope to use cloned pigs to grow organs that can be transplanted into humans.
Okay. By the way, they have already started doing that. All right. So we've got up here some DNA technology and biotechnology. So these are just medical processes that involve doing things like cloning and genetic engineering. And so I want you to, so hopefully you've read through these. And one of the big projects is the Human Genome Project, which has actually gone through and mapped out all the possible genes that are in a human being. And they have studied them, and they use this project so that they can make these manipulative gene switches that you saw in the first one, recombinant DNA. So recombining characteristics. DNA fingerprinting. This is very current these days in solving crimes. Genetic engineering, like the one that you just saw in the recombinant DNA, so that you can create the characteristics that you want to have or remove the characteristics that you don't want to have. And genetically modified food. This is in your grocery store that they're making not only just organisms, but a lot of times when we think of organisms, we think of the fish and chickens and things like that, but also the plants are being genetically organized. You know that seedless foods are not a normal thing. So if it's seedless, that means it was genetically modified because it was created not to have seeds. So you have to keep on grafting and doing technology to keep reproducing that, because seeds are the mechanism by which fruits and vegetables normally reproduce. So if it's genetically modified, then a lot of times it's where they've removed the seeds from it. All right. So what I want you to look at, so again, make sure you've given that page a read. What were the activity that you're going to look at? I'm going to do this from the student view. To make sure it's there. All right, so I'm looking at this with student view. You're looking for the DNA technology poster. All right, so you are going to decide, and you can research the different types of DNA technology, and you pick one, just one. So this is not a overall view of all the technology that's available. You pick one, and then you want to use this rubric. You're going to create a poster. And your poster needs to have all of this information in your poster. So you can do this as a Google slide. So you're not doing a paper poster, although I guess you could do a paper poster and then um, uh, take a picture of it, but you can do this as Google Slides. All right, so you're going to do a brief summary of whatever technology you selected. Okay, then you're going to, provide details about the DNA technology work that has been done. You want to discuss, and notice that one's worth 30 points. So you want to give a lot of detail as to what's being done with that technology. All right, what are the human benefits of this technology? Are they just doing this for science research, or what is it good? What is it going to be good for? I don't. Okay. And then I apologize for that, because being this is my first year, I don't have previous posters to go from, but this did come out of the biology assignment folder that, so it's been used before. I didn't make this up, but they did not leave examples for me to look at. All right. So again, you want to research, pick a type of technology, define it, then describe all the techniques that they do, kind of like the video on cloning just did. They explain to you, and you can use cloning as one of the technologies, explain exactly what are steps they take in order to create a clone. So that would be your 30 points worth. What benefit is this to human? So so benefits. So going down the side for criteria is what you really want to follow. And then you want to make sure that you are trying to get the max points out of it by looking at the first column, not the last column, because you don't want to lose any points. All right, so human benefits from resulting from this type of technology, possible bioethical issues. There's been a lot of controversy about cloning, for example, playing God. Same thing with recombinant DNA. They controversy there too about people who genetically modify their fetus so that the baby looks like what they want it to look like. So there's a lot of controversy about that. So this question here about bioethical issues, it revolves around, should we or shouldn't we, are we playing God, kind of thing. Next one, visual effects. So you want to have pictures, you want to make, it's a poster. So make it a good looking poster. It shouldn't just be black and white with words on it. Make it look like a poster. Nice title to it as far as what technology you've chosen. And then maybe have, boxes and corners and things to your poster so that it's neat. Evident artifacts, presentation, again, just be creative. This one on digital poster. You can do a video if you have that capability. You can do, and actually, you can do a slideshow. It just shouldn't be a long slideshow presentation. If you're more comfortable doing a slideshow presentation, that's fine. If you have so much information that you don't fit it on one page, that's good too. We are going to share these, so you're not just doing it for me to see, okay? And have references. So you should have a reference slides separate to the poster, please. Okay, so that you don't have to cram it all into the one space. So I'm thinking that a Google slide presentation probably would be better, but it should not be very many slides. It should be 3 or 4 slides long, okay? You can break that down into a title slide and then a slide that defines your technology with a picture. Then you can have a slide that has the next to the human benefits and bioethical. That could be one slide together because that's not a lot of information. And then everything else is just pictures and neatness. Okay. So use the rubric to help you um, decide maybe I'll go back and put that in there. Let me go back and edit this to so that so everybody's going to be neat about this. Let's make that. Okay, we're not going to just have a poster. We're going to do Google Slides. It should be, and your, okay, use Rubik to create a DNA. Okay. Okay. All right. And in your information slides, it should include pictures. Okay, we'll leave it like that. All right, so we'll make it that, and that should make it easier for everybody. It should make it so that you're not cramming everything into one space. You can spread things out. It just should not be 20 slides long. It doesn't need to be any longer than, yes, you can. And let's do that. So Isabella's already asked, can she do cloning? So cloning is gone. Let's everybody choose one, and that way we'll have some variety. So take a minute. All right, so Isabella's got that. Nobody else do cloning. Somebody else, just keep typing it and send it to everybody and not just to me. Okay, so when you choose to send the message, send it to everybody. Okay. So pick a technology, tell me what it is, do it fast so that nobody else is doing yours. All right, Piper's doing fingerprinting, so nobody else do fingerprinting. And I'm writing this down. There's only 8 of you, so we're good. Gene therapy. Natalie's doing gene therapy. And again, we're going to have what we call a gallery walk, so everybody will get to see everybody else's activity. So this is why it's good that everybody does something different, so we have something else to look at, and everybody's not doing the same thing. All right, so we got 3. All right, so if you picked yours, go ahead. You can go ahead and get started working on it. There's things that we're doing these days, ancestry DNA, all that kind of thing. Somebody could do that. All right, so don't forget, pick one and tell me what you're going to pick. So I got four of you that haven't chosen anything yet. Okay, so I got Malik, Maxwell. All right, Maxwell, Ariana, Malik, Kashawn. Have y'all picked anything yet? Talia. Okay, genetic engineering. Okay, gotcha. All right, so again, um, you should have a title slide, and that can have pictures on it. Kashawn, DNA sequencing. Okay, gotcha. Your name should be on your title slide. All right, still waiting for Ariana and Malik and Maxwell. Vaccines. Okay, Malik, I got you. And that one should be interesting because of all the vaccines we're doing now with COVID and flu shots, and you always have to keep changing them because the viruses adapt. Okay, nobody's doing genetically modified organisms. Maxwell, Ariana, either one of you, want to jump on that? All right, so the due date on this is all the way, we're off all next week. It'll be due the following week. When you come back on Tuesday, we'll do a review of this unit on DNA. Wednesday, we'll do a quiz. And this is the last unit we're doing before final exams. So after that, we'll just be reviewing the semester's work. So it is a full semester exam. So again, our DNA unit quiz will be the Wednesday that you come back. We'll review Tuesday when I see you. Take the quiz on Wednesday, and then this is due the Friday. And then after that, we will not do any new topics. Your exam is actually, you're the last exam. Fourth period is, so we have quite a few. So anytime that you're not in exam, we have time to review. So fourth period is December 17th. So we have plenty of time to review this semester and do activities for review. And again, that will be a full semester exam. All right, Ariana and Maxwell still don't have a answer from you. All right, you got paternity testing. That's how they do paternity tests, genetic engineering, so to speak. Like I said, ancestry.com, paternity tests, those are things, topics you can choose. GMO, nobody has any of those. Okay. Hmm. Isabella, you can do both. Talk, you can say how it started and then switch to humans. That could be part of your how does it affect human section. Mhm. Mhm. Okay. If you need any help or clarity, just let me know. Ask questions, please. Okay, folks. You all have a great Thanksgiving. See you in December.